So in this first video, we're going to be talking about pyruvate processing. So the purpose of glycolysis and the citric acid cycle and the electron transport chain is to produce ATP. And we're going to look at part of that pathway today. So we've already talked about glycolysis, in which we took 6-carbon glucose and converted it into 2 molecules of pyruvate. And pyruvate, each molecule has 3 carbons. And in this process of turning glucose into pyruvate, we generated 2 NADHs and 2 ATPs. The next step we're going to talk about is pyruvate processing. So pyruvate processing takes place inside an enormous enzyme complex called pyruvate dehydrogenase. So that's the enzyme that's really working during this reaction. And what pyruvate dehydrogenase does is it converts pyruvate into acetyl-CoA. Okay, so pyruvate undergoes a series of reactions. One of its carbons is oxidized to CO2. So here we're releasing one of the carbons. So CO2, we have a carbon coming off. NADH is produced. So with every oxidation reaction comes a reduction reaction. And the remaining 2 carbons are attached to the coenzyme A molecule, producing acetyl-CoA. It occurs in the mitochondrial matrix, this step right here, in eukaryotes, and in the cytosol, and in bacteria and archaea. Okay, so what is the mitochondrial matrix? So here's the mitochondria, and the mitochondria inside have these cristae, which are these sac-like structures, and inside the cristae is the intermembrane space, right? And outside of the cristae here, which is the area outside of those sacs, is the mitochondrial matrix, okay? And so if this represents the membranous stacks that form the cristae, inside, let's say each line here represents that double membrane, inside that, or outside of that, but inside the mitochondria is the mitochondrial matrix. And this is where this reaction takes place in eukaryotic cells. So pyruvate processing is regulated by feedback inhibition. So we're in each process, in each one of these steps, we talk about how the pathway is regulated. And remember, in glycolysis, we had regulation, feedback inhibition of phosphofructokinase. Now we have regulation, feedback inhibition of pyruvate dehydrogenase. So when products of glycolysis and pyruvate processing are abundant, pyruvate dehydrogenase is phosphorylated. And this phosphorylation of pyruvate dehydrogenase changes the shape of the enzyme complex, and then it becomes inhibited. So this is an example of when phosphorylation, instead of activating an enzyme, inhibits it. And so when the reactants are abundant, pyruvate dehydrogenase is then dephosphorylated and can go back to converting pyruvate into acetyl-CoA. So far we've talked about glucose being converted into pyruvate, which is converted into acetyl-CoA. But in the bigger picture, right, this is starting with a carbohydrate glucose, fats and phospholipids as well as proteins can come into this pathway here, this catabolic pathway, and enter in at acetyl-CoA. So to summarize what we've gone through today in the process of pyruvate processing, we've converted pyruvate into 2 molecules of pyruvate into 2 molecules of acetyl-CoA, and 2 molecules of carbon dioxide came out. We also produced a net of 2 NADHs, since 1 NADH was produced in each one of the reactions. Okay, this NADH is going to eventually go to the electron transport chain, and we're going to use it to make ATP, but we'll talk about that for the next class. And the enzyme responsible is pyruvate dehydrogenase. This is regulated by inhibitory phosphorylation during feedback inhibition. So we can turn this off when there are a lot of products, and then we can turn it back on when there aren't a lot of products. So the next reaction I'm going to talk about is the citric acid cycle. But that's all I have for this video.